Hello, everybody. This is our applied paper for this week, which takes some of the concepts in the lecture. And we can see how those concepts relate to some aspects relating to resource conservation, environmental protection, sustainable harvest, and related sorts of things. So this week we've been talking about sexual systems. And that simply means how the male and female function, the sperm and egg producing functions, are combined or separated or used for reproduction within individuals of any species. And we talked about hermaphroditism and diesi. Diesi is the separation of sexes into separate individuals, and hermaphroditism is a sexual system that combines the two sexes in one individual in various ways. And sometimes it can be simultaneous, in which both eggs and sperm are produced at the same time, and an individual can be a father or a mother with a single copulation. And in other cases, hermaphroditism is sequential, and individuals can start out as one sex and change into another sex. And the timing of the change can depend, or the signal, the trigger for the change can be variable. It can be from outside the individual, somewhere in the environment, in the social environment, for example, or it can be a signal that comes from inside the individual. A certain size, for example, could trigger the change, or a certain mass threshold could be passed, and an individual can change sex then. And this can happen in invertebrates, vertebrates, and also plants. And this paper by Schramm and Steele, published last year, takes a look at the effects of fish harvesting, size selective fish harvesting, on the sexual system of a sequentially hermaphroditic reef fish. This is called the black-eyed goby. So this species is a protogynous species, which means individuals are born female, and after a certain period of time, they change sex into males. Now, they chose a protogynous species, but they could equally have chosen a protandrous species to look at these effects, and people have done both. So, just to give people, give you, a context to think about this, I'm going to show another couple videos of sex change in one protogynous species and one protandrous species of fish. So, we'll start out with a clownfish, which starts out as a male. They're all born as males, and after a certain period of development, they will change sex into females. So this species is a protandrous species.
My name is Justin Rhodes, and I study behavioral neuroscience. We've chosen a model organism that shows a very dramatic example of social environment having a long-lasting impact on the behavior and physiology of the organism as a model system to understand how the brain orchestrates this. And these are clownfish, which change sex depending on the social environment. We have clownfish paired off in 26 tanks. One male, one female, and maybe a couple of other smaller undifferentiated fish living on sea anemones. And the larger one is always female, dominant. And the second in the hierarchy is the male. And if you remove the female, then the male will change sex and become female in a matter of a couple of months. And basically, this is through the brain interpreting this social change. The clownfish has both testicular tissue and ovarian tissue, and so it's a matter of just encouraging one of the other tissues to develop. It's unclear exactly what signals they're using, probably mostly vision, and maybe some olfaction, and maybe even sound, and then that probably communicates to the hypothalamus in areas of the brain that we know communicate to the pituitary gland, which is a little gland right below the brain, which releases hormones into the blood, which communicate to the gonads, causing testes to absorb and ovaries to develop and morphology to change and completely reverse the behaviors, because this used to be a male and now it's dominant and behaving like a female and so on. One hypothesis is that a sex change will involve some reorganization of the brain, and we can measure and track when we remove the female and observe what changes take place in the brain of that male changing sex into female. We can see what areas of the brain are sprouting new neurons or what areas of the brain are removing new neurons. We can discover then how, in this one dramatic example of how the social environment reorganizes behavior and presumably makes some inferences. The two human implications potentially are humans also have a similar, they don't change sex, but social environment can have long-lasting effects on their behavior, personality. Presumably this involves similar biology. Another potential implication of this work is identifying changes in the brain that apparently are necessary to function as a female versus a male. If you measure male and female brains, you see differences. The question is, which of those differences are functional? What are the neuroanatomical or physiological differences between a male and a female brain that makes males behave like males and females behave like females? Difficult to know when you compare males and females in different species. But in a sex-changing fish, presumably those changes that are actually undergoing reorganization, new neurons sprouting up in one area, neurons degrading in another area, those are most likely to be functional, because that's going to be very energetically costly to the animal to start rearranging the brain and in connections and so on. I mean, think about what's happening here. The fish is just detecting sensory, a change in the social environment, and somehow that's affecting basically the testes to be changed, ovaries to develop, the animals to get larger, their behavior to change, genitalia is changing. So this is a really remarkable example of how your experience, your social experience, can have long-lasting effects on your behavior and personality and morphology, really, anatomy in this species.
I just want to make one point here. The trigger for sex change in the clownfish is not anything endogenous to the individual. It's not an internal signal. It's an external signal in its social environment. So the sex change is caused by removal of a dominant female from a male's immediate environment. And when that dominant female is removed, then in a few weeks or a few months, that male will then respond to that removal by changing sex to a female, as described in this video, with all the accompanying changes in the brain and body and behavior. Okay, so that's our example of a protandrous species. Now, a video of a protogynous species, the bluehead wrasse.
In coral reefs off the coast of Florida, a species of fish called the bluehead wrasse is helping scientists understand sexual development. Researchers are interested in a striking change the small yellow females undergo in their social groups. When the larger dominant male in the group leaves, the largest female changes sex and takes the male's place just days later. Scientists have documented sex changes in many species of fish, but they still don't have a complete picture of what molecular mechanisms drive the change. To help figure that out, researchers from Australia, New Zealand, and the US removed dominant males from groups of bluehead wrasse in the waters off Florida, then analyzed tissue samples from the remaining fish as the sexual transformation was in progress. What they found is that the transitioning female starts behaving differently within hours after the male is removed, and the fish's coloring becomes darker as it eventually develops the distinctive blue head of a male. To investigate how these changes correlate with changes to the fish's genes, the researchers analyzed gonad cells from the transitioning fish. They found extensive shifts in gene activity, and in the pattern of methyl groups attached to the fish's DNA, a modification that can affect the function of certain genes. For example, genes associated with the production of female hormones turn off. As estrogen levels collapse, there's a gradual increase in genes responsible for producing androgens, hormones associated with male reproductive activity. Ovarian tissue also starts to degrade and is replaced by testicular tissue, which starts producing sperm within 8 to 10 days. The researchers think this genetic rewiring might get triggered by increased levels of cortisol, a stress hormone that spikes in female fish soon after the male disappears from the group. The team hopes a better understanding of how cells get reprogrammed in this tropical fish might help scientists understand how genes are turned off and on in other animals, and even humans.
So in both of these species, the trigger for the sex change was the social change, the elimination of the dominant individuals in the immediate population, the local population. It wasn't a size threshold or a weight threshold of an individual that triggers the change. Okay, so what's the problem? Well, the problem is if you have a sequentially hermaphroditic species and you have a fisheries that targets a specific size of that species, like a minimum size limit or an intermediate size limit, then you will have an effect on the sexual system of that species because the sizes are correlated with sex and sexual behavior and mating behavior. And so that fishery will create artificial selection on the sexual system of that species, artificial selection that could cause evolution of a different sexual system. It could cause evolution of differences in the growth rate, of maturation, the time that a fish matures to be able to reproduce from juvenile to adult. It could affect the triggers of sex change, and it could affect the reproductive output, the total reproductive output. If you're affecting the sex ratio, if there are fewer females, then the population has a lower reproductive output. And these all could be permanent evolutionary changes in the sexual system of the species caused by the fishery. Do you want to be causing permanent evolutionary changes in the sexual system of your commercial fish that is the resource that you're depending on? Well, if you do that, it might cause collapse of the species because it no longer becomes a sustainable harvest. All right, so these authors wanted to test the hypothesis that a size-selected fishery might affect the growth, maturation, sex change, and reproductive output of this species. And they did this by a great field experiment where they went into the field and selectively removed about 25% of the fish that were established in the populations on these artificial reefs. And they were simulating a pulse of fishing mortality with, for example, minimum size limits or slot size limits or maximum size limits, which are used in many fisheries. So their predictor variable was fish removals, size selective fish removals. Either they were removed or not. That was their predictor variable. Their response variables were growth rate, maturation, sex change behavior, and reproductive output. Important to point out, though, that the black-eye goby is not a harvested species. It's just a species that's common and easy to work with and might respond in a way that's similar to other harvested species in the area. So perhaps it could provide insights in how a harvested species could respond. So what did they find? Well, so they had basically 4 treatments. There's a control, the large removal, large fish were removed, medium-sized fish were removed, and small fish were removed. And so these are the results: number of individuals, the density of individuals in the population, male, female, and juvenile. And this just shows that the removals had the intended effect. The large removal, males were less abundant in the large removal because you removed males. In the medium and small removal, the females were less abundant than controls. In the small removal, the juveniles were less abundant than the controls. So the removals had the effect that they wanted it to have. All right, so what effect did the removals have on this response variable, mean growth? Well, no significant effect. Here's the control: large removal, medium removal, and small removal for male, females, and juveniles. You can see by the error bars that there were no significant effects of the treatments on any of the three sex types. And the error bars are pretty wide. These are only one standard error. Remember, the 95% confidence interval would be double these. So some of these would extend almost all the way down to 0. So there's a lot of noise in this experiment, as is true in general for field experiments in ecology. Look at the small sample size. Overall, no significant effect of the treatments on mean growth. No significant effect of the treatments on proportion of juveniles that matured. No significant effect of the experimental treatments on the eggs produced by females over time. Large, medium, and small removal. Okay, so what do you think? Do you think that size-selective harvesting is okay? That we can conclude that species are resilient to size-selective harvesting? And that harvest won't affect the growth or maturation or sex change or reproductive output of any of these protogynous hermaphroditic reef fish? Well, maybe and maybe not. There are a lot of questions that we could still ask and that are generated by this study and its design that I'm sure you folks can think about and will think about. All of these concerns are certainly not resolved by this study, and there is still a lot of controversy and concern about this type of harvest. And to be fair to the authors, they did point out that there is an alternative interpretation of these results, namely that our experiment lacks sufficient statistical power to detect biologically meaningful differences among treatments. You know, normally you think that a paper that doesn't find any significant differences is difficult to publish, but I think these papers should be published. This is also important information that should be out there, and I'm glad this paper was published, even though they did not find any significant differences in their treatments. It was a really nice field experiment, manipulative field experiment. If they had a higher sample size, they might have found significant differences. We don't know that. And the authors do point out that we also need to study other factors that could be important as well. Harvesting intensities and details of the reproductive ecology. And, you know, as our knowledge improves, then we can more effectively manage commercial species that are sequentially hermaphroditic. All right, that's our paper for this week. Read it and learn from it. See you next time.